Hello, I'm Andrew Lee, an Omnilogic Fellow and an intern at Columbia University this summer. My project is coding an app in R using single-cell RNA-seq fetal brain samples for the identification of potential autism risk genes. Autism is a neurological condition that causes limitations in social communication, repetitive and compulsive actions, and also declines in cognitive ability. There is a high prevalence of autism, as around 1% of the total population has this condition. The similar clinical symptomatology of neurological disorders also leads to common misdiagnosis of autism. This is a problem because it serves as a barrier for the creation of accurate individualized treatments. And also, occupational therapy programs and learning arrangements depend on accurate diagnosis. The current methods of diagnosis are the DSM-5 and genetic testing. The DSM-5 is considered the gold standard diagnostic criteria used by physicians today. However, several studies have indicated that the DSM-5 is overly broad with its criteria. As you can see on the right, this is a flowchart of how autism is diagnosed, from the behavioral monitoring to the genetic testing. And the genetic testing is highly dependent on discovering new autism risk genes. Recent studies have estimated that 1,000 or more genes cause autism. However, there have only been 100 genes that are confirmed. A more recent approach is through machine learning to identify genes that have similar expression profiles to already confirmed autism genes. For my project, I want to first identify potential autism risk genes using an unsupervised machine learning approach. Next, I want to identify those genes that have similar expression profiling among autism-related cell clusters. And this would eliminate some of the coincidental connections caused by the unsupervised methods. Next, I want to improve my research and the research of others by creating a publicly available app with a user-friendly interface that only requires a single gene name to result in clear visualization and plots of more than 20 types of human cells. My dataset for this was an ARIS dataset from Chen et al. 2020. There were two groups of data. First was the human fetal brain sample readings, and the next was the prefrontal cortex samples. For the identification of possible autism-related genes, I first conducted a Heatmap and principal component analysis. The plots were annotated with around 200 genes, and 121 of those were from the Spark Autism database. The app was coded in R using Shiny and Shiny dashboard packages, and some of the code was modified from the Omnilogic Biotech transcriptomics course. To continue with the methods, I first used a t-distributed stochastic neighbor embedding plot, the t-SNE plot. And next, I also did violin plots to use single gene inputs to generate expression levels for more than 20 distinct cell types. And the next was a ridge plot, which is just an alternative to the single gene t-SNE. The expression levels were then compared with those of known autism risk genes, and the genes with similar expression profiling were determined as possible autism risk genes. So you can see a schematic of how the process was conducted. First, this used an expression matrix, and then the heat map and principal component analysis with hierarchical clustering was made. This gave me a list of genes to further analyze, and this I manually inputted on the app that I created. And this created violin plot, t-SNE plots, and ridge plots. And this I compared to the known autism risk genes to develop a final list of candidate autism genes. For the heat map and principal component analysis with hierarchical clustering, you can see that the risk genes are clustered near the top with high functional scores, and these are genes that have already been evaluated as autism genes. However, 10 genes listed were not already labeled as autism genes, and they were also clustered near the top, and you can see this with the purple and blue coloring. And this means that they can potentially serve as candidate autism risk genes. You can see a similar expression profiling on the PC1 of the principal component analysis. The autism risk genes in blue are grouped together. However, you can see several red dots, and these indicate that these genes are also similarly expressed. These genes from the heat map and principal component analysis were listed for the next step in verifying that they are indeed risk genes. To narrow this down, I used a single gene expression from the app I coded. All the genes from the previous step were manually inputted one by one into the coded app, and they generated 3 plots, as you can see from the example CHD4, which is a known autism gene above. You can see that CHD4 had high expression in many of the cell clusters that are linked to autism, and also in terms of the scores where they are on the t-SNE plot, you can see that it is highly expressed in many cell clusters. And this process was verified for each of the genes, and if they had 1 low expression levels, or 2 were not expressed in autism-related cell types, they were excluded. On the right, you can see the cell type plots, and these were the UMAP and also the complete gene t-SNE. While they do not help in autism risk identification, you can see that the cells can be clearly grouped into 26 distinct cell clusters. My research conclusions is that the app provides automatic plot rendering and image downloads, and this means that the user can easily visualize single genes. The app is also useful for other researchers that can want to correlate genes of interest to expression levels between cell types. For this study, I correlated the application to autism risk genes, and you could see that on the heat map and the principal component analysis, there was apparent clustering of the known autism genes and also on the heat map clustering. However, there were also some genes that were not known as autism risk genes before, but were also grouped with these. And after applying the 2 selection criteria, 5 genes were identified as potential autism risk genes. These may also be markers of autism. On the right, you can see the list of the 5 genes identified. The discovery of autism risk genes can clearly improve the genetic testing available for autism, which provides more clear and accurate diagnosis. In the past, genetic testing has been limited by the lack of autism risk gene knowledge and the amount of datasets available. For some limitations of my study is that brain expression similarities can often be coincidental, and further functional analysis is needed to verify that any of the genes are indeed autism risk genes. Supervised machine learning approaches and also neural networks can provide more autism risk gene discovery and reduce the risk of false positives. For my future research, this analysis has proved useful in identifying the 5 candidate autism genes, but I also want to study their biological and molecular functions. To extend my project further, I want to continue adding more recent and larger datasets to use in the app and the unsupervised gene machine learning methods. I would like to thank Mr. Elia Brodsky, Dr. Harpreet Kaur, and my mentors at Columbia University. Thank you for listening.